how it can be used for manipulation of the plants. As we have seen, or as we are observing in the diagram, Agrobacterium tumefaciens bacteria, the TI plasmid is isolated, and in this TI plasmid the gene of interest, which may be of a other plant, is inserted into TI plasmid. Then it is again sent back into the bacterial cell. Which bacterial cell? Same, Agrobacterium tumefaciens. And this Agrobacterium tumefaciens cell will enter through the wounds or the cuts into the plants, where it will integrate with the plant genome. As a result, the plant genome from one plant will be expressed into the another plants. So this is how the TI plasmid is able to carry along with it the gene of one plant to the other plants, and when it is expressed, it imparts certain characteristics of the foreign gene, like a disease resistance or weedicide resistance, etc. So this is how these vectors are able to carry the host target gene from one cell into the another cells. So friends, we have discussed about various kinds of vectors in depth. There are certain vectors for animal cells also. For example, retroviruses, because these retroviruses are able to infect the animals and integrate with them. Baculoviruses, these are the two viruses which are widely used for vectors, or they are widely used as a vectors for animals. Now, because of the time constraint, we are not going to discuss about these two viruses or the vectors which are used for animals, but basically the principle and the techniques remains the same. તો મિત્રો, તમને આજે ડિટેલની અંદર વેક્ટર્સનો શું રોલ છે રિકોમ્બિનન્ટ ડીએનએ ટેકનોલોજીમાં એ આપને હવે ખ્યાલ આવ્યો હશે. તો આ સાથે આપણે આપણું ડિસ્કશન અહીંયા એન્ડ કરીએ છીએ, અને જો આપને કોઈ ક્વેશ્ચન્સ હોય, કોઈ ક્વેરીઝ હોય તો આપ પૂછી શકો છો. આ સાથે હું આ ઓપોર્ચ્યુનિટી લઈશ કે ખાસ બાયસેકનો અને ડિપાર્ટમેન્ટ ઓફ હાયર એજ્યુકેશનનો, કમિશનર ડિપાર્ટમેન્ટ ઓફ હાયર એજ્યુકેશનનો આભાર વ્યક્ત કરીશ, because they have been the one which are responsible for starting such a unique lecture series, SANDHAN, through which we are able to reach to the students. I would also like to thank the staff of BISEC Studio, who have taken a keen interest and presented this discussion to you in an excellent fashion. On behalf of Kiran Ben and on my half, I would like to say thanks to all of you. Thank you very much. Namaskar.